we'll be discussing things that go on below the neck in neuroscience. Not my area of expertise. I'm usually used to above the neck neuroscience, but we have a set of 3 interesting talks on the spinal cord. We'll start with Felix Fiederling, who is a postdoc in the Dodd and Mason labs at the Zuckerman Institute at Columbia University. You can find him at Felix Fiederling on Twitter, which we'll put in the chat box if you want to go ahead and follow him. He'll be presenting his work on 3D mapping of the whole mouse spinal cord. Take it away, Felix.
Cool. Thank you very much for the introduction and also being involved in organizing this meeting. It has been very much fun so far. Yeah, and as you said, I will talk about tools that we made to analyze the spinal cord in its entirety. Okay. So the spinal cord is a quite critical part of our nervous system and allowing us to engage with our environment by relaying and processing somatosensory and motor signals. And research on model systems like the mouse has identified major classes of spinal neurons that establish these functions. However, as you heard throughout this meeting and with recent advances in transcriptional profiling of spinal neurons, it has become clear that we have massively underestimated the complexity and heterogeneity of the spinal cord. So to achieve a better characterization of spinal cell types and the detailed functional circuitry, we need to map the spatial relations of neurons and projections in the spinal cord with better precision and most importantly in relation to other neurons. So ideally in the context of the entire spinal cord. However, currently we don't have the tools to do this and classical histology approaches are quite impracticable because the spinal cord is so long, right? And we also lack still a common coordinate framework for spinal cord analysis, making it complicated to map data with precision and to compare data from different samples and across groups. So while we have powerful tools today at hand to identify spinal cells and to trace their circuits, there is a need for an easy to use and high throughput method to analyze the whole spinal cord and to map the positions of cells and projections into a common framework. So today I will talk about how we address these issues in 2 basic steps. So first of all, how we created tools to make the collection of data for the whole spinal cord more efficient. And then secondly, about how we develop software to automate the analysis of spinal cord sections in 3 dimensions and to map the data to a reference atlas. And so starting with the first problem, we first thought for ways to make the sectioning of whole spinal cord more efficient. And one way to reduce the work and time involved in this process is to split the tissue into multiple tissue segments and embed them in parallel in the same block for synchronous sectioning. So as shown here, by splitting the cervical to lumbar region of the spinal cord into 9 tissue pieces, we can bring down the number of actual sections that we need to cut from over 1,000 to about 100. However, now with these small tissue pieces, it is quite tricky to embed them in the same block in a perfect orientation without confusing their identity. So to help embedding, we developed spine racks, and spine racks are 3D printed support structures that can fit into an embedding mold and help guide tissue pieces into place. And so similar to a wine rack, they hold objects of interest in a preferable orientation. These spine racks, we made them from PVA. This is a polyvinyl alcohol, a water-soluble material that can be softened through incubation in OCT embedding medium after the tissue has been placed and before freezing the block for smooth sectioning through the material. PVA is actually a component of OCT, and after sectioning, any remains can simply be washed away by washing in PBS. This material is commercially available as filament for many 3D printers, and so it's very easy and cheap to produce spine racks in large quantities and also in different geometries for other applications. So using these spine racks, we can collect all sections of the spinal cord within only 1 hour of sectioning time and on only 15 slides, which means a massive improvement in time for both sectioning and also subsequent immunohistochemistry compared to traditional approaches. And in addition, The precise arrangement of tissue sections on the slide also makes it possible to image the sections automatically on a slide scanning microscope like this, while keeping track of the identity of each tissue section. So to address the second issue and to analyze the section data now in 3 dimensions, we developed a processing pipeline for Fiji that we call Spinal J. And this pipeline follows these basic steps shown here that I will explain in a minute, and with exception of the first step, runs fully automatically after initial setup. The overall hands-on time, including the first step, is currently a half an hour to an hour, depending on the size, the section quality, and also the size of the data set for one animal. So I can't go into much detail, but this first step basically of the pipeline is required to bring the individual tissue section images into a rostrocaudal order, as shown here, and then to sort out empty and damaged sections that are still included, and to come to this and to orient the sections so that we end up with a continuous stack of intact sections that is ready for image registration and 3-dimensional reconstruction, as shown on the right here. And for this registration process, either neurotrace, which is a fluorescent Nissl stain, or DAPI can be used. But besides of that, you can of course stain for whatever you like. So now, after we have created this 3-dimensional reconstruction of our section data, let's see how we can bring this data into a common coordinate framework and to analyze it in the context of anatomical annotations. And because there is no 3-dimensional atlas for the mouse spinal cord, we have built one from the 2-dimensional Allen spinal cord atlas that consists of 34 annotated sections, one for each spinal level, that were drawn based on Nissl stainings. And so to do this, we first digitalized these annotations of the Allen atlas by filling all the regions with a color code, and then we built a 3D volume from both the Nissl and the annotated sections, simply by stretching the sections of each segment to match the rostrocaudal length of each spinal level, as has been measured by others. So this new atlas does not add any rostrocaudal resolution compared to the Allen atlas, but rather brings the segment annotations into rostrocaudal context and into a 3-dimensional format. And this now allows us to map our experimental data first to the Nissl reference using either the neurotrace or DAPI channel, and then overlay the 3-dimensional annotations for analysis in atlas space. So, and as shown here, Spinal J is able to map sections shown in magenta to the Nissl template in green with very high accuracy, using either neurotrace or DAPI on the right for registration and mapping. We have also quantified the mapping accuracy of neurotrace signal to the gray matter annotations and find across multiple animals an average accuracy of about 90%, confirming the visual impression of good mapping. So after establishing the basics, let's talk about some experimental data. And for this, we first labeled corticospinal neurons by injecting a tomato AAV into one side of the cortex and then mapping their projections into the spinal cord. And using Spinal J, we can quickly reconstruct the sections and analyze the mean intensities of signal per atlas region, as shown here in the middle. And this is a summary of the data for each segment. And this allows us to quickly assess the distribution of signal across the entire spinal cord. And this on the right is a zoom in. We can also combine individual atlas regions into more intuitive groups of regions, for example, gray matter laminae or white matter tracts. And this allows us to compare the distribution within these broader regions, for example, also across hemisegments, left versus right. And in our analysis, we see that within the white matter, the signal is mapped to the dorsal funiculus, and within that region, specifically to the corticospinal tract, as we would expect. And in the gray matter, the distribution of signals matches quite nicely to what others have reported at cervical level using manual approaches, with the difference that, of course, now our data set can also be analyzed at different spinal levels. In Spinal J, we also have options for automatic cell detection and detection of projections, including a tool for machine learning pixel classification that is called Elastic. And as shown in this example here, where we genetically labeled FOXP1 expressing motor neurons, we can train Elastic to recognize either the soma or the projections, as shown in red and green respectively. And we can then use these probability masks to separate cells and projections from a single image channel and analyze their respective densities in each atlas region. This segmentation approach is also particularly useful when dealing with signal in a high background, as shown here in this example of an IB4 staining of non-peptidergic DRG fiber terminals. And as you can see, compared to the intensity analysis shown on the top, the analysis of the segmentation of the segmented projection densities gives us a much clearer result, showing that these fibers innervate lamina 2. So in my last slide, I want to focus on how Spinal J can be used to compare data across multiple samples. And for this, we mapped FOXP1 positive cells from 4 different animals, and we plotted their positions in a different color for each animal here on the left side. And you can see that the distribution of cells from the different animals aligns quite nicely without any obvious color shifts that would indicate major mapping offsets. And when we look at the number of cells in each spinal segment, we see that the cells are equally distributed across animals and that they concentrate at limb levels as we would expect for this type of motor neurons. We also calculated the distances between the centers of mass of the individual cell distributions in each animal and within each segment, and find that the average mapping offset across all segments is around 30 micrometer. So very, very small offset. And this really shows that we can map cells in different animals with high accuracy, and that allows to directly compare these samples. So, and with this, we now have quite a powerful tool, I think, to analyze the positions of all different types of cells, projections, and even synapses in relation to each other. And this allows us, maybe as a community even, to build a detailed map of the entire spinal cord. We're currently finishing up Spinal J and make it available for everyone shortly. But in the meantime, please also feel free to contact me if you're interested in using SpineRex or Spinal J. And so with this, I want to end by thanking the lab, my mentors and collaborators, especially Luke, who did most of the coding for Spinal J, and of course, all of you for your attention. Thank you.
Thank you, Felix. Nice talk. And right on time, great timing. And so feel free to ask questions in the Q&A. I'll start. So in your last slide, you showed very good consistency in terms of registering across individuals. What strain of mouse did you use, and do you expect there to be differences across different strains?
So we looked at some cell populations that we labeled across different strains, and we don't see major differences, but maybe this is also because if there are any differences in size, maybe of the spinal cord, these will get compensated by mapping it to this common framework, right? So yeah, so the problem at the moment is that the atlas is not very accurate. This atlas is built from a single mouse, right? So the annotations, I think, have to be improved by building an atlas that it comprises several animals, and then you can talk more about inter-animal variability.
Thanks. Any other questions?
Can I ask a question?
Of course, speakers are free to ask questions as well.
Yeah. So Felix, awesome work. I have been missing this for a long time.
Yeah.
Is it possible to use Spinal J to align one individual section? I mean, I have so many pictures of sections of the spinal cords. Can I just use the program to find where it would sit?
So for robust alignment, at the moment, it's still the best if you can tell it what the rostrocaudal extent is of the volume that you want to map. In regions where we have a lot of morphological landmarks, so if you have like motor neuron clusters that clearly identify a certain level, it can do it automatically. But for example, in the thoracic cord, where even the annotations for several segments are the same, there's no way really to map it precisely unless you tell them where it should go.
Okay, thank you.
But and we did this the spine regs, you can certainly use them not only for the analysis of the whole cord, but sectioning maybe from 9 animals only a lumbar fraction of the cord and then map those volumes.
Yeah. Nice.
Thanks again, Felix. We'll move on.